Hello everyone. In this video, we discuss about signaling through G protein coupled receptors. This one has three essential components: receptor, G protein, and an enzyme. Here, receptor is with 7 transmembrane helical segments. You can see here. The 7 alpha helices are connected by loop of various length. 3 loops outside. And these 3 loop together to form ligand binding site where the signal binds. And there are 3 loops inside, 3 loops on the cytoplasmic side. And this G protein bind to third intracellular loop of this receptor. And examples of GPCR receptor are beta adrenergic receptor, serotonin receptor, rhodopsin receptor, etc. that we can study next class. And next is G protein. What is G protein? G protein is guanosine nucleotide binding protein. And G proteins are heterotrimeric because G protein consists of 3 different polypeptide subunit. Which are the alpha, beta, and gamma. The guanine nucleotide binding site is present on the alpha subunit. The G protein binds either GDP or GTP. When GDP is attached to this alpha subunit, the G protein is inactive. And when GTP is attached to alpha subunit, then this G protein is active. And here you can see the G protein is attached to the plasma membrane by lipid chain and that are covalently attached to alpha and gamma subunit. And the third component is an enzyme. This enzyme in the plasma membrane generates intracellular second messengers. Examples of enzymes: adenylyl cyclase, phospholipase C, and cyclic GMP phosphodiesterase. Now we can study about the signaling mechanisms. First, signal binds to receptor. What happens? The signal binds to receptor. Then there is a conformation change occurs in receptor. And this receptor bind to G protein. When the receptor bind to G protein, what happens? It induces G protein alpha subunit to release its GDP. And it is replaced by GTP. Here GDP is replaced by GTP. And the GTP binding to this G alpha subunit causes a conformation change where in the alpha subunit. And what happens? The G protein released from the receptor and it also triggers the dissociation of GTP bound alpha subunit from the G protein. First, this G protein is released from the receptor, then the alpha subunit from the G protein is released. And the released alpha subunit then bind to enzyme. And it activates the enzyme. The activated enzyme produces second messengers. Now we can discuss the transduction of epinephrine signal. Here the signal is epinephrine. Epinephrine is a hormone and it is otherwise known as adrenaline, fight or flight hormone. And when it is produced during a stressful condition. And epinephrine causes an increase in blood glucose level. How it breaks down? It induces the breakdown of glycogen. For what purpose? To provide extra energy to deal with the stressful condition. Epinephrine is produced during a stressful condition, and epinephrine induces the glycogen breakdown to provide more glucose in blood. Epinephrine increases the blood glucose level by breakdown of glycogen. Here, signal is epinephrine. Epinephrine bind to the receptor. Here, the receptor is beta adrenergic receptor. Where the beta adrenergic receptors are normally located? On muscles, liver, and adipose tissue. Then, as we discussed earlier, this beta adrenergic receptor bind to G protein. Here, the G protein is of GS type. GS. G proteins are of four types: GS, GQ, GI, and G12/30. So here the G protein is GS. The beta adrenergic receptor bind to G protein and the GDP is released. GTP is attached to G alpha subunit. Then the G alpha subunit dissociate and binds to the enzyme. And here the enzyme is adenylyl cyclase. It releases second messengers. Which is the second messenger here? Cyclic AMP. This cyclic AMP activates protein kinase A. Protein kinase A activate phosphorylase B kinase. Phosphorylase B kinase activate glycogen phosphorylase kinase. And this glycogen phosphorylase kinase convert glycogen into glucose. This is about epinephrine signaling, GPCR signaling mechanism. Now we can discuss questions. This is a previous year question. The question is, the G protein coupled receptor consists of three protein subunits, alpha, beta, and gamma. And in unstimulated state, the alpha subunit is GDP bound and inactive. And in stimulated state, the alpha subunit is GTP bound and it is active. In order to regulate this G protein activity by regulating GDP and GTP concentration, the alpha subunit acts as option 1 GTPase, option 2 GDP kinase, option 3 cyclic GMB specific phosphodiesterase, option 4 cyclic AMB specific phosphodiesterase. Here, the inactivation of G protein is controlled by its GTPase activity. The G alpha subunit can turn itself off by hydrolysis of GTP into GDP and phosphate. This results in conformation change, causing a decrease in the affinity for enzyme and increase the affinity for beta and gamma subunit. Thus, following the hydrolysis of GTP, this G alpha subunit dissociate from the enzyme and reassociate with the gamma and beta subunit to reform inactive heterotrimeric G protein. So the answer is GTPase. The second question. This is also a previous question. The second messenger cyclic AMP synthesized by adenyl cyclase transduces a wide variety of physiological signals in various cell types in mammalian cells. Most of the diverse effects of cyclic AMP are mediated through activation of protein kinase A, also known as cyclic AMP dependent protein kinase. Which of the following statement regarding this protein kinase A is not correct? Here, this is the structure of pKa. First, this one is inactive R2C2 tetramer. It contains two regulatory subunits, and these are empty cyclic AMP sites. 4 empty cyclic AMP sites, and it also contains two catalytic subunit, but this one is inactive. Here the substrate binding site is blocked by autoinhibitory domains of R subunit. This is the substrate binding site. Substrate binding site is present on the catalytic subunit, which is blocked by autoinhibitory domains of R subunit. Then cyclic AMP binds to regulatory subunit. When the cyclic AMP binds to regulatory subunit, what happens? This autoinhibitory domains buried. It causes a conformation change, and the catalytic subunit dissociate from the regulatory subunit, and this became active pKa. This is open substrate binding site. So we can check these options. Inactive pKa is a tetramer. This one is inactive pKa. Is a tetramer consisting of two regulatory R subunit and two catalytic C subunit. So here the option is correct. Second option, each R subunit bind the active site in a catalytic domain and inhibit the activity of catalytic subunit. Yes, this option is also correct. Each subunit, third option, each R subunit has two distinctive cyclic AMP binding site. Yeah, each R subunit has two cyclic AMP binding site. And binding of cyclic AMP occurs in a cooperative fashion. And this one is also correct. And so which one is incorrect? The fourth one is incorrect. So the answer is this one. If you like this video, please subscribe and like. Thank you.